next presenter, which would be Margareta Lakusic. And she is now a PhD student at the Research Center of Biodiversity and Genetic Resources in Portugal. And before that, she studied molecular biology and neurobiology and is involved in a lot of topics from population ecology up to conservation of reptiles in Serbia and North Macedonia, where she's come from. Early this year, she was awarded with a grant for establishing a long-term monitoring study of the endemic and highly endangered meadow vipers, Vipera ursinii macrops in Serbia. And looking at her publication list, it's revealed that she is highly interested in even further snakes of the Balkan region. And so we are happy now to hear more about her current work about Balkan vipers, story of the venom evolution and conservation, chapter 1.
Thank you very much, Mike, for this quite brief introduction. And thank you all the organizers for having me and inviting me to give this talk about my current research and things that I will be doing in the following 3 years during my PhD. And also I will give you a short story about the currently conservation projects that are happening in Balkan Peninsula. So let's start. First, we all know what is a snake venom, but the things that are important to point out is since it evolves multiple times across different taxa, we can say that this implies that it is a key evolutionary innovation. And then knowing that by its composition, it is a mixture of enzymatic and non-enzymatic components, and their snakes are using it to overpower prey and for the defense. I like to keep always in mind that this trait is a link between snake as an organism and its environment. And always when I'm thinking about snake and snake evolution, this is something that I'm coming back to it and thinking why it evolves. So knowing this is a functional trait, and we can say that this is fastly evolving phenotype, and it is dictated by different factors. So one of the main factors that is causing the venom evolution is the diet. So meaning that venom most often co-evolves with the physiology of the prey species it is used against. But also important things to point out is that this variation can occur on all taxonomic levels. So between different species, of course, venom differs greatly. But then also when we look at the level of the species, going at, for example, between different populations, also the venom is changing differently. If we enter the population level and look just between different individuals, for example, between males and females, some expression profiles may differ. Then if we take only one individual and follow it through the time, also the changes of its venom from the neonate, juvenile, and adult state, it is different. So one more interesting and important thing that was recently discovered is also the fact that snakes are using the venom for defense. Therefore, also this behavior is altering in a way the venom composition. So this is a broad story of how snake venom involves in all taxa, but I'm going to talk about the genus Vipera and the Balkan vipers. So a brief introduction of this would be that there are around 30 species currently recognized of this genus. They are inhabiting quite a diverse habitat, so they can be found in the dry, rocky grounds to all the way to the more humid, more vegetation-rich environments, and also different types of altitudes. As for their distribution, they are spanning across Mediterranean region, and also they are found all the way to Central Asia. But when we think just about Europe, southern peninsulas, so Iberian, Italian, and Balkan Peninsula are actually the hotspots of diversity of viper species. And then let's take a short journey to Balkan Peninsula and talk a bit why is this area such important and interesting to study. Well, this region of Europe is one of the richest areas in reptile species. It is caused by the rich geological background in a way that different corridors were appearing and disappearing. There were dramatic changes in climate, dramatic changes in habitats. So there is a high level of endemism in this area. And also, as other peninsula, Balkan Peninsula was a glacial refuge throughout the Quaternary glacial period, meaning that species that were found refugium here are highly diverse between each other. So then when we are now in Balkan Peninsula and thinking about the vipers there, the most notable species here is the nose-horn viper. So this species is widely distributed across the whole peninsula. And this is, I would say, a great model species for studying the venom evolution because, first of all, this is highly genetically diverse species. It is represented with 7 distinct clades only in Balkan Peninsula. Also, there are differences in diet. So between males and females, there are noted differences in diet. And also there is an ontogenetic shift with different types of prey used by juveniles and adults. Another important thing, is that venom of nose-horn viper is highly complex, and it is also, with having the wider distribution, this means that also there is a high incidence of snake bite with potentially lethal outcome, meaning that this species is classified in the category 1 and 2 of the medically important species by the World Health Organization. So knowing all this, it is not strange that this venom has already been studied to quite some extent. So here we can see venom profiles of different subspecies, and just taking a look at it, it is clear that there is this venom is highly variable, and there are some interesting differences between these subspecies that are also belonging to different genetic clades. But in most to some extent, again, some aspects are neglected. So, for example, the age, sex, and phylogenetic relationship was, at some extent, was not really taken into consideration. So, with this, I would like to study the venom variation more, putting it into ecological and genetic background. And this will be done by studying the variation at the intra and interpopulation level, along with studying the venom variation across phylogeny. So for the intra and interpopulation level, I would say that there is a great model system to study this question. So here is Golengrad Island, also known as Snake Island, found in North Macedonia, and it is a place of high. So the name Snake Island it got by the high density of, first of all, die snakes, of vipers, and also there are high abundance of Hermann tortoises. And there is one conservation project currently ongoing on this island. So if you're interested in seeing what is actually happening, and a lot of things are happening on this island, you can check it in social media. But for my story, why it is super interesting to study the venom evolution here. So this is the unique situation where nose-horn viper has quite a different diet than any other population found on the mainland. So on the islands, vipers are preying mostly, almost exclusively, on lizards and centipedes. But however, there is an ontogenetic shift as well here. So juveniles are preying mostly on centipedes, while adults are including or almost exclusively feeding on lizards. So here the idea is to collect the venom from all age classes, also to collect the venom from different sexes, so for females and males, and then potentially there is an opportunity to also collect the venom from pregnant females in order to see if reproduction is also somehow alternating the expression of certain components in this venom. And then in order to see how similar or different it is, we will also collect the venom samples from the mainland population. So it is quite good that in the near proximity of this island, there is a population with a normal situation. So they are having the broader diet range. They are including also the small mammals. So here we will generate the proteome of the mainland population, and then we will compare all the venom venom profiles from the island to this mainland population program. Important thing to mention also here is that this population belongs to the southern clade, because this will later be included in this higher level of study. So my idea is to study also the venom across phylogeny and to see how any phylogeny is actually affecting the venom composition. As mentioned, there are 7 distinct clades on Balkan Peninsula, and with stars you can see the populations where we will collect venom samples, and then we will perform the venom on a chip analysis, which is I think good protocol to study this large-scale comparative analysis. And the idea here is, yes, to see if the venom diversity is consistent across phylogeny, meaning that we want to see if the genetically closer groups will also have similar type of venom composition. And then, if not, also to see what are the factors that are affecting this change. So this is some of the main points of my main focus of my PhD that will going to be developed in the following 3 years. But there is one also side project that I am now working on, and it is the conservation of meadow viper in Serbia. So the meadow viper is one of the smallest European vipers, and this is one of the most endangered reptile species in Europe. So they are asked their habitat and climatic factors that need to be fulfilled are quite narrow. They are under constant pressure of habitat loss. And in Serbia, there is the subspecies Vipera ursinii macrops, which is found in the alpine and subalpine meadows above 1,000 meters. And so far, we actually don't know what is the conservation status in Serbia. So what we knew is that there are 2 localities in near proximity where meadow viper was in some moment noticed. So the idea was to actually check whether in these localities meadow viper still exists, then to search for additional suitable areas where meadow viper can occur, and also to see which are the threatening factors to these habitats, and also directly which can influence these species. So our fieldwork in 2021 led to discovery of a new locality where this species is found. And this means that with this locality, we are increasing the area which will be suggested for protection, which is really, I think, important considering the status of this species in general. And also in the following year, we will have the opportunity to do more field work and to study more which are the most important threatening factors and most present threatening factors, and how to overcome or to reduce them. So this is a small conservation project, but since this is a venomous species, I was also interested, and I am interested in studying the venom of this species. Since it is so rare, this species has already was studied recently. So just in 2020, Langbalia and authors have identified 25 different proteins and generated the proteome. So comparing to other vipers, this venom is much less complex. But going back to the diet, why this species could be really interesting to study? Because comparing to all the other vipers, this meadow viper has quite distinct and different type of diet of feeding preferences. So during the most active season, it is preying on orthopterans, but in some population, there was shown that at the beginning, so right after hibernation, they are also choosing small mammals. So knowing that this situation in one population in Italy, I was thinking that maybe it would be interesting to see if there is also a seasonal shift in diet in the population in Serbia. So the other subspecies, but still alpine meadow species. And then to see if this seasonal shift also maybe leads to seasonal shift in venom composition. So this is something that maybe will be done in the future, but it is something that could be interesting to study more. And with this, I would like to thank you for the attention and to open the talk for questions and discussion, suggestions, whatever there is. I'm still now starting everything, so any insight, any comments are highly valuable.
Thank you, Margareta.
Thank you.
Really nice talk. And I'm always happy to see European snakes in presentations. And Tim asked, how prevalent is gene flow between Vipera ammodytes subspecies, and does it have an effect on your sampling strategy?
So no, at least in the places where we are sampling, there is no gene flow. Potentially in some northern populations, there was noted that there is gene flow between Vipera berus and Vipera ammodytes, but in all populations where we are sampling, there is only Vipera ammodytes occurring.
I think his question was more about the subspecies of ammodytes, if there is a gene flow between them.
Okay, yes, I did not return the subspecies. Yeah, good question. At the in the hybrid zones, in the just mere edge of the subspecies range, there could be the gene flow, but again we are choosing the populations where there should not be any gene flow between subspecies nor between clades.
Okay, thank you. The next question: do you plan to do you plan to sample across ontogeny on the island and in the other population that you're looking at?
So yes, for sure in the island I will collect the samples of juveniles and subadults and all age classes that I can find. Also in the mainland population, the idea is to sample as well the venoms of the juveniles in order to compare, to actually see, for example, if the venom profile of juvenile from the island is the same, or it is in a way it's really similar to the venom profile of adults, because if they're not changing diet, this could be the outcome of our analysis. So at least in two populations I will collect the venom samples of juveniles and see how is the changes across ontogeny.
Okay, thank you. Do we have any other questions?
I mean, that's it. I've got a sort of semi-serious question, which is, can I go to Snake Island? Because it sounds very cool.
Yes, next year there will be more. It is super nice to see, actually, because there are different processes that are ongoing. Also, story about tortoise is super interesting. So different stuff can be worked there. It is nice to see.
Well, I think if that is, there's one from Wolfgang. I think he's just saying you should look at ontogeny throughout. It'd be interesting to see what happens in the dwarf populations in the Aegean Islands and whether they show some kind of neoteny. So
that would be the dwarf population. But I think that here, if there is some kind of neoteny, it would be driven by the constrictions in diet. So because they don't really have any changes in diet, well, not any, but not dramatical changes in diet. I'm not sure what is the situation in these Aegean islands, what is their feeding behavior, but I will look into that. Thank you.
We've got another question. Are you planning on doing functional assays with meadow viper venom, especially because they're eating arthropods?
So at this moment, no, because at this moment I'm focusing on my PhD, which is on Vipera ammodytes, and I'm collecting samples of Vipera ursinii. So if there is, in some moment, opportunity to work and maybe do the functional assays, I would probably consider that. But for this, at this moment, I cannot say anything more about the meadow viper venom.
That is fair. I think if that's it for questions, then it's about time to wrap up our session for today. I'd like to thank our three speakers again very much for agreeing to talk with us, and for you all for listening and joining us today. That's any ado